has contaminated the R bacteria and turned them into S. Witchcraft.
General for the transfer.
So is it possible that when
I
you
mean,
marry
statistics will probably
into culture,
or
you can almost say that the remaining skeleton of the dead bacteria culture merged with
But you
our
all
bacteria
irids
culture
in
allows
nature.
certain traces of DNA that weren't denatured during the heating process to enter through the permeable membrane of the R bacteria, thus making its way to our bacteria's nucleus and essentially transforming it.
Mm-hmm. Great. Exactly. So the idea is that there's this transforming principle. They didn't know what it was, but they knew that if you take S bacteria and you heat it up until it's dead, you don't have living cells anymore. All you have is a soup of chemicals, right? It's just a mixture of chemicals, all the ones we've talked about so far. The carbs, the fats, the phospholipids, the DNA, the RNA, the protein, all of those just sort of in a soup. And so by taking that skeleton, as you described it, and combining that with the living R bacteria, something is able to come into the R bacteria and carry information, right? So this transforming principle is information. And we know that dead bacteria are just a chemical soup, so that needs to be some kind of that essence, that information is chemistry, right? Yeah.
So then, what would be the potential, I guess we could say, the ceiling for DNA before it pyrolyzes and basically becomes carbon?
So if you, DNA is remarkably resistant to being broken apart like monomer from monomer by heat. So you can actually heat DNA up really, really hot, and the two strands may separate, but you won't necessarily hydrolyze the bonds that hold together, that is, break all the bonds that hold the monomers together. So it's surprisingly resistant to that.
Okay.
Which is one of the reasons you can actually, in 50,000-year-old fossils, recover intact DNA. So it's incredibly resistant to being broken down over time, as long as the conditions are correct. Right? So how would you go about, what would be the next step you would take if you have this situation where there's this chemical stew that can carry information and convert one bacteria into another type? How would you go about sorting through that chemical soup to figure out what the chemical is that actually controls the heredity? Sure.
You would look through, you would take, after the process was over, you would just like dissect, I guess, the parts, the different parts of the soup to find out which parts are consistent within both, or is that all on the right track?
So Julian's saying dissect apart the chemical stew to find the parts and see if they still, tell me if this is what you're saying, still have the information needed to convert R into S.
Yes.
So if you take that stew, which is the carbs, the phospholipids, all of those different parts, and you find some way to separate them out into the different parts and then test each one separately to see, is it actually still able to carry the information? And that brings us to the next experiment, which is this Avery et al., just means, and others, because there are a bunch of people who participated in this, where basically what they did is they did what you're describing and purify different components of the cell, right, of dead S bacteria, and then use it to see, can it still turn R into S? So I've given you sort of a simplified, slightly fabricated, but that's okay. This is just to test your thought process, chart that shows you a process that they undertook in these Avery experiments where they took the dead S bacteria, they eliminated lipids, and then tested to see, does it still transform R to S? So look at this data set, all three of the conditions, and see what you think this supports in terms of which molecule carries the information.